So our lab has created a couple of tools, HIV Toolbox versions 1 and 2, for integrating all the readily available information about HIV proteins into one place that's easy to use and see everything about them in relation to each other. In the main, after you've chosen one of the proteins, in HIV Toolbox 1 there's a window here where you can select further information you want to view. You can select different structures of HIV that we have, protein interaction sites, functional sites, short functional motifs, and anything you select in the upper left corner sequence window here is automatically loaded into the actual three-dimensional JMOL viewer windows below and on the right. The first one displays the motifs in this protein and domains, or short functional motifs and domains in the protein. The middle one is the protein-protein interactions and functional sites. The third window is for homology. Our purpose here is to incorporate information about sequence, structure, function, and evolution. So in the homology window, you can kind of see how the different residues have been conserved from 0 to 100%, depending on what you select. And then in a new version, the windows on the right here, we have drug-resistant mutations, where you can view different types of drug-resistant mutations for this particular protein that you selected, and the different drugs that cause them, and the drug binding sites in the bottom window for the drugs for that protein. So on the drug-resistant mutations, we came up with a classification scheme to color them in and categorize them. We came up with 5 different basic types of mutations. A primary mutation, which is colored red, is a mutation that will confer resistance to a drug all by itself. Primary set is a group of mutations that will confer resistance to the drug, but only if they occur together. A beneficial mutation will undo or counteract drug-resistant mutation, primary mutation. A beneficial set of mutations, if they occur together, will counteract primary mutations. And a secondary set will counteract primary, or will enhance the effectiveness of primary mutations. And what we have displayed here are the FDA-approved protease inhibitor drugs and the active sites on protease, the binding site for darunavir, and then in the lower right corner, the conservation of the amino acids involved here. And some interesting patterns that you wouldn't necessarily be able to see easily otherwise can occur. For instance, there's a residue here that's near, you can see it's near the active site over here, and it's near the drug binding sites here. And it does different things in response to different drugs. For atazanavir, part of a beneficial set mutation. For darunavir, it's a beneficial mutation all on its own. And for falciparenavir, it doesn't seem to do anything. For indenavir and lopinavir, it's part of a primary set. And for novinavir, it's like a primary mutation all on its own. And it would be more difficult to determine this if you didn't have the program capabilities that we created here, just to see everything in relation to each other all on the same screen all at the same time. And you can kind of see in the conservation window that particular residue is a lot less well conserved than a lot of the residues of protease here. And what we've done with this information, we came up with a tentative plot of resistance mutation estimates for different countries for all the different drugs. These are the same protease inhibitors mapped out into a world map that I showed on the last page, but you can do the same thing for any of the drugs that are FDA approved. And you can see we're hoping to make it a clinical tool that is useful in a case where you wouldn't have the genome for a particular patient's infection. For instance, in Japan, the resistance to atazanavir and lopinavir is very high, but the resistance to other drugs is less so. And so if you were a doctor who didn't have genome information for your patient, you might want to avoid those 2 drugs just as a matter of course. And we wondered if maybe the resistance matched by subtype that occurs in the different countries, the prevalence of subtypes. But when we compared it to the distribution of subtypes by geography, it didn't turn out that way. So we're still, this application is still in alpha stage, it's still being verified, and we need to check it, but we've got HIV Toolbox 1 and 2 and the primary version of this application up on our website, and these are the URLs here at the bottom here. And so you can go there and visit it and use the site right now.